Thanks for including me, and thanks to the other speakers for some great talks. I'm going to talk about essentially some alternatives to mapping. If you want more details for what I'm up to, of course, you've already mentioned the book After Phrenology, but there are a couple of recent articles that I'm just listing here. So anyone who's interested in following up on the details of what I'm up to, these are a couple free places to look. So here's an outline of what I'm going to talk about. I'll talk about some basic brain facts. I'll go through that quite quickly because actually a lot of the main points have come up already in the first three presentations. And then I want to talk about componential explanation in neuroscience and the way in which it does kind of assume a mapping relationship. And I'll argue, however, that much of the data that we're seeing suggests that that kind of mapping assumption might not be where we want to go in the neurosciences. So I'll propose very briefly that instead of componential explanation, that we think in terms of constraints and what I'll call enabling constraints. And then I'll talk more in general about what some implications of that are. First, some basic brain facts. So there are at least two kinds of neuroplasticity in the brain. The more familiar one is Hebbian plasticity, right? The phrase neurons that fire together, wire together. This is the up or down regulation of individual connections between individual neurons in the brain, obviously crucially important to all sorts of neural functioning, learning, and whatnot. But there's also another kind of neuroplasticity that I've been calling neural reuse. And what neural reuse does is establish multiple overlapping neural coalitions for different purposes. So the same parts of the brain might be involved in several different kinds of tasks, but they will cooperate with different partners in each of those tasks. So the point here now is that reuse leads to a very particular neural architecture where, as we've heard already, the mapping between cognitive processes and brain regions is many-to-many. Individual regions of the brain are used and reused in diverse tasks. And then differential behavior, differential function, is the result not of different parts of the brain being active, but instead of dynamically combining the same neural elements in different. So here's just one example of the notion that individual parts of the brain. I want to orient you to these plots for a minute because they're important. So these are called spider plots. In the way I'm going to be using them, each of these vertices indicates a particular category of psychological function. So in this particular case, although I'll show you other examples, we have emotion, action, language, working memory, attention, and so on. And then the distance out along the axes here indicate the relative proportion of activity in a particular region of the brain in that category of psychological function. And it just turns out, and I won't go into the empirical details here, but it just turns out that most regions of the brain are highly functionally diverse. So the anterior insula here, this is this one, the 0.88 one, that's a especially diverse region. And as you can see from the spider plot here, this particular study used 11 different cognitive categories, cognitive functions, but it's active in literally every single one to some degree or another. A more typical region of the brain is here, this 0.76. That's an area of left thalamus. And you can see it's active in most of the regions, but to a lesser degree than anterior insula. And then a quite atypical region of the brain is this here, auditory cortex. Auditory cortex turns out to be a relative specialist in the brain. And so it's primarily active, almost entirely active in auditory tasks, and with a little bit here and there in a couple other tasks. But it's, you know, that is actually a specialized region, and there are specialized regions in the brain. But as you can see from the histogram over here, right, there is very little in the brain that looks like auditory cortex. Let me give you just one more example. So this is a a study that used quite a bit more fine-grained categories. And these are the 6 divisions of left and right insula. And as you can see, even though these are very related regions, each part of insula has a very different functional fingerprint. You've heard that a phrase already in the first talks. The idea here being that each part of the brain is different from each other part of the brain. There's definite functional differentiation. But for a long time, we took that differentiation to indicate functional specialization, and that inference does not seem to be warranted by the actual data. And as an example of the changing partnerships, this is just an illustration of the functional partners of left precentral gyrus, in this particular case in semantics tasks. And this, again, this is a meta-analysis based on a couple thousand. And I'll show you that same region of the brain and its partners in a couple of different tasks. So this is semantics, so things having to do with linguistic meaning. This is emotion, various emotion tasks. And this is stimulus-driven attention tasks. So let me just go back through those again, because just to get the idea, right? So the same region of the brain takes this set of partners when participants are dealing with tasks with linguistic meaning, this set of partners in various emotion tasks, this set of partners with stimulus-driven attention tasks. And so the idea is, right, each part of the brain is involved in many different things, set of partners under each condition. This is an example, as it says here, of large-scale neuromodulation. And one of the super important things we need to understand about brain function is that neuromodulation is ubiquitous in the brain, and it's ubiquitous at multiple spatial scales. So here's a slide that'll come back, orienting you to this. This is a study of individual neurons and the networks it participates in C. elegans, the soil nematode that we have a complete neural mapping of neurons. So ASH in red there. You see that in all the different categories here. So it's involved in various behaviors, often all avoidance, feeding. It's involved in aerotaxis, that is finding and following oxygen. It's involved in various social behaviors, including aggregation and repulsion. But in each of these cases, just again focus on ASH for the moment, that red neuron there. In each of these conditions, it's actually synapsing with and or cooperating with a different set of partners. So SH is involved in all these different behaviors, but is involved with different partners in each. And when we look at neuromodulation that far back in the evolutionary history, that's a sign, presumably, that we should expect to see neuromodulation as a crucial element of brain function across all taxa. There's a different version of neuromodulation, so something called volume transmission, in which basically neurotransmitter gradients across the cortex, or not just the cortex, but across the brain, change the relative likelihood of activation in various structures. Anyway, so that's the idea. We've got these many-to-many mappings, right? Individual regions of the brain, multiple high-level uses. That function is achieved by putting the same bits together in different configurations. And there's significant functional differentiation and significant functional integration. What I want to suggest is that these facts and some things that I'll mention in a bit challenge what is perhaps the leading account of explanation in neuroscience, which is componential. Here's just one quote. This is from Carl Craver. So what is componential mechanism? Well, it construes explanation as a matter of decomposing systems into their parts and showing how those parts are organized together in such a way as to exhibit the explanatum phenomenon. And a somewhat different take from Craver and Bechtel. Scientists discover lower levels by decomposing the behavior of a mechanism into the behaviors of its component parts, decomposing the behaviors of its parts into the behaviors of their parts, and so on. And so the idea here, and I think, you know, as has been hinted at in the talks already, this is really at the root of the mapping assumption here. The idea is that we can find the parts that have a particular function, break that function down into component functions, find the parts that execute those component functions, and repeat until we ground out. So we'll call this decomposition and attribution. And it's important here, and it will be important to see the challenge to the view of some particular functional relationships in the brain in a bit. That function then emerges from interacting structure in a strictly bottom-up fashion. We find the parts that execute very low-level functions, and we see how those parts interact with higher-level parts, and so on, and that gives us eventually the phenomenon, psychological phenomenon of interest. So, if we're going to satisfy the norms of economic explanation, we have to be able to identify the phenomenon of interest. They call it, identify the system that does that thing, identify the relevant spatial subparts, I call that the set X, of the mechanism M, and their relevant properties or behaviors, and then describe how the parts in the set X are organized such that the system does its thing. One example that Craver works out in some detail is the action potential, right? So the phenomenon is the action potential of the neuron. The system that executes that function is the neuron itself. The parts in virtue of which the neuron executes the action potential include elements of the cell and its surrounding ionic milieu, the positively charged potassium and sodium ions, gated ion-specific membrane channels, blah, blah, blah. We don't need to go into detail here, but the idea is that we identify all the parts, we figure out what the function of those parts are, we see how those parts are organized together so that we end up with the phenomena of interest. So here's starting to get to the main point here. I think the ubiquity of neuromodulation puts pressure on this account, in part because it assumes identifiable subsystems with fixed behaviors or functions. Right? It assumes we can come up with simple attributions of function to spatial subparts. But it's not clear that that's really a warranted assumption or idealization. So that first spider plot there is dorsal anterior insula, left dorsal anterior insula. If we want to identify the behavior of left dorsal anterior insula, well, which behavior do we have in mind? It's clearly active under a huge range of circumstances. Similarly, what's the behavior of neuron ASH? Right? It's involved in a number of behavioral functions and a number of circumstances. Which behavior do we have in mind? Now, obviously, we could save componential mechanism simply by weakening their requirement to something like, well, let's identify a behavior fixed at a given time or in a given context. And there have been suggestions along those lines. But I think maybe it's time to rethink this issue in a bit more systematic way. One motivation is, as I say here, because it begs the question of how those component functions get temporarily fixed in the first place, even assuming they are. And I think it's a fair assumption that at a time, a given part of a given neural system has a thing it's doing for a period, and then it probably changes its function. These spider plots, by the way, these are network fingerprints. And what these exhibit are the combined functional fingerprints of the parts of each of these networks. And I'll put this here to make a particular point. So just look at that particular network there. That's the dorsal attention network. It is at least purportedly a functionally specific network, right? Specific for a particular kind of task-oriented attention. look at the functional fingerprint, and you'll notice that it does not consist of attentionally specific parts. It consists of parts that are active in audition and vision and executive control and imaging and so on, right? So we have a purportedly functionally specific arrangement of parts, the parts of which are not themselves functionally specific. So there's a significant question here of how you move from non-specific parts to a functionally specific organization. And there are other kinds of suggestive potential issues with both the mapping assumption and componential mechanistic explanation. So here's one, a very small example, but I find it quite telling. So a study by he, recent study by he, finds that stimulus-driven and intrinsic activity negatively interact. That is, there's less response to the same stimulus when background activity is higher. Why is that important? Well, that means it's difficult to identify the impact of a given stimulus, the impact, right? What does a stimulus actually do? Trial averaging won't recover the true estimate. And so what is it that a stimulus is actually causing? Well, if it changes depending on what the background activity is, there's a significant issue here in trying, well, to move forward in terms of functional attribution. You know, put straightforwardly, our search for the function of a given part then gets hindered by this limitation. What a part does depends on the context within which it's responding, and I think this leads and this may go back to Joe's talk in a certain way, leads to context dependence of accurate functional attributions. Right? What we should say a particular part of the brain does is going to depend on the larger context. Here's one very particular example. So this is a review by Randy Buckner of hippocampal place cells. Hippocampal place cells famously are cells that fire when an animal is at a particular region in a particular space. Turns out, not surprisingly, that, of course, they're more complicated than that. So section B here indicates the firing of a particular hippocampal place cell when the animal is at a particular location. This is the famous kind of function that these cells have. But it turns out, and the wave you see in the background is the overall fluctuations of background activity of the hippocampus. And it turns out that it has its classic function of firing when an animal is at a particular place, only when it fires in sync with the reduction in background activity of the overall hippocampal organization, hippocampal activity. It fires also when an animal is just about to enter a particular location, but it does so at a different part of the background fluctuation wave, the gamma, I think it's gamma band fluctuation. It also fires when the animal has just left a location, and it does that at a different point in relationship to the overall background activity. And I think, again, this puts pressure on the notion of the very notion of components, right? The whole idea of a component is a identifiable subpart of a system with a very particular output. But if the output or behavior of a part of the mechanism is different based on background conditions of the larger mechanism, what hippocampal place cells do is fire in sync with gamma fluctuation when an animal is at a place, and either a little bit before or a little bit after that fluctuation in other circumstances. So if the input-output mapping is unstable in this way, or depends, as I say, on what the background conditions are, it's not clear to me what the claim to identify a component comes down to. What's a component function under these circumstances? And moreover, then the dependence of function on background conditions raises the possibility that there may be a violation of the bottom-up assumption that Craver and others have so strongly. That is where one identifies lower-level parts and then shows how they interact to give rise to higher-level function. I'll give you a very particular example of this possibility. So here's a picture of a Starburst Amacrine cell. You can see why it's called a Starburst cell. These are really interesting cells. So these are found in mammalian retina. They're an important part of our motion detection. There's a cell body, as you can see in the middle, and then dendrites. There's no axon for this cell. An array of dendrites arranged around that cell body in the kind of format you see here. What's interesting about these cells is each dendrite is direction selective, right? So each Starburst amacrine cell dendrite is a directionally selective spatial subpart of the overall cell. This is how you describe this system from the componential mechanistic viewpoint. And what that means basically is that the dendrite will release neurotransmitters. They're very funny cells, right? So dendrites release neurotransmitters in this case. They don't just receive input, they actually have output. So when motion, so say light moving across the retina, goes in the direction of the dendrite itself, the dendrite will release neurotransmitter, thus signaling that it's detected motion in that direction. An important thing to know about Starburst amacrine cells is they are in a very densely interconnected and overlapping network, as you can see here. Why is that important here? Well, it turns out that what makes Starburst amacrine cells directionally selective, and I won't go into too much detail here because the point is simple. The mechanism is complex, but the point is simple, is that you get bipolar cells. These are the light detecting cells in the retina. Bipolar cells synapse onto these dendrites so that when you get light moving across the bipolar cells, they are synapsing onto the individual dendrites in such a way that will cause it to fire, to release neurotransmitter. At the same time, those bipolar cells, when they go across a neuron, a dendrite in the anti-preferred direction, that dendrite will actually inhibit the dendrites of the neighboring cells. Right? So what you've got here is a set of a network arranged such that you have mutual inhibition between neighboring cells. And it's actually this mutual inhibition that causes the cells, the dendrites, to be motion selective, direction selective. If you take a Starburst amacrine cell in isolation from its larger network and you run light across it, all of its dendrites will actually release neurotransmitter in response to that motion. Right? So removing it from the network actually destroys the direction selectivity of the dendrite. So whereas it would be natural to explain direction selectivity in terms of some kind of intrinsic lower level properties and spatial subparts of the dendrites, this looks like a case where it's actually the opposite is true. It's the organization of the network that explains the direction selectivity of the dendrites, and not the dendrites that explain the direction selectivity of the cell. Right? So the mechanism here appears to be both extrinsic, not spatial subparts, and at a higher organizational level than the system that we're talking about that exhibits the important and you know, for our motion detection systems, crucial phenomena. Just to make the point further, there are also something called direction selective ganglion cells in the retina. And these actually inherit the direction selectivity from the SAC dendrites, right? The SAC dendrites selectively synapse onto these direction selective ganglion cells and suppress their responsivity. And that suppression of responsivity actually leads to the selectivity of the direction selective ganglion cells. So there's actually no intrinsic mechanism inside the direction selective ganglion cells for motion detection, right? That direction selectivity is imposed on those cells by the Starburst amacrine cells, those dendrites, and those dendrites get their direction selectivity because of the way they are organized in a larger network. So look, I think these kinds of examples in the organization of neurons warrant an amendment, at least, to mechanistic explanation. That is, the parts of mechanisms in virtue of which systems exhibit behaviors are not always spatial subparts or lower-level components of those systems, right? They sometimes operate via the imposition of functional constraints from whatever spatial level. Let me just go into that possibility in slightly more detail. So just reminding you, here's a situation with something even as low level as a single neuron in an animal, in a behaving animal, ASH, participating in multiple kinds of contexts. So what I want to say about what I'm calling enabling constraints is, look, ASH has the same capacities in all its networks, right? It has a set of things it can do. It can fire in various ways under various circumstances. But capacities, in this sense, are necessary conditions, but aren't yet functions. They aren't yet behaviors of that component. So, for instance, direction selective ganglion cells, in the absence of Starburst and Amacrine cells, continue to exercise their capacity to fire in response to light, but are strictly speaking non-functional, right? If we take their function to be indicating specific direction of motion, they don't have that function. They have the capacity to fire in response to light, which they will exercise, but they do not yet have their assigned function in the retina. It's only when they're constrained by the Starburst Amacrine cell dendrite that they end up actually having their function. So constraints in the brain limit capacities so as to enable function. To make it slightly more formal, you could say that an enabling constraint is a physical relationship between a functional system and a set of entities at the same or a different level of description, such that the set of entities changes the relative probabilities of various possible functional outcomes of activity in the system. As explanations, I think enabling constraints have a couple of interesting advantages. First of all, they're synchronic. So whereas cause is this one thing happens, then the next thing happens, an enabling constraint is synchronic, right? It's an imposition of constraints at a time. Also potentially symmetric, that is, there can be mutual constraints. It's not as if you have to identify as if you do in cause, the asymmetric relationships here. It also easily accommodates various kinds of inter-level explanations. You know, Craver is famously kind of allergic to top-down causation, but that's because it's top-down causation. You can certainly have top-down constraint without violating any kinds of epistemic or other. And it applies at multiple spatial scales. And what I want to suggest, and this is something that is amenable to both theoretical and empirical investigation, and this is one of the things that I'm working on these days. I think understanding things in terms of constraint may well help us explain how selectivity emerges in large-scale organizations of non-selective parts. But the idea is quite simple. You'll have realized this already. The idea is if you can imagine each of the parts of a large-scale network mutually constraining one another, then you could sort of imagine that essentially what's happening is the relationships between these parts are selectively enhancing or inhibiting various capacities of those parts, with the result that the organization as a whole can exhibit concerted and very specific behavior. Right? Parts of the network interact so as to impose mutual enabling constraints. We might be able to selectively inhibit and enhance some subset of existing capacities, and that could lead to organizations, to mechanisms, to networks with specific functions, even though, strictly speaking, they're not composed of functionally specific parts. So let me just then kind of round this out for a minute here by relating this to the notion of a synergy. So a synergy is a functional grouping of elements, could be neurons, muscles, limbs, individuals, temporarily constrained to act as a single coherent unit. There is in the brain mounting evidence that one effect of task-related stimuli is reduction in the overall variability of neural activity. That's important here because it's a well-established finding that reduction in the overall variability in the parameters defining a system is a sign of the dynamic construction of a task-relevant synergy. So one particular example of which and it was studied extensively by Bernstein, who was he studied motor control and motion. When you look at a blacksmith hammering a part, it turns out that there is more variability at each of the joints of the blacksmith's arm than there is in the trajectory and the strike location of the hammerhead. The idea here being, right, that the variability in each of the parts is acting as mutual constraints on each of the other parts, so as to maintain that hammerhead in the right trajectory and strike location. Right? So the arm of the blacksmith has become a synergy in that sense. We see something similar in the brain, whereas, so the idea here is along various functional dimensions here. They're just showing three. When you're off task, the state of the brain can be in any of those red places, right? You can see a kind of a large cloud of possible locations in a functional space. When the brain is on task, it instead occupies that blue region, a much more constrained location in a functional space, right? This kind of finding suggests that when a person enters into a particular task, one of the things that happens is their brain forms or becomes a kind of synergy and occupies a very small region of a functional space, which is what allows the possibility for concerted effort or concerted attention to or concerted performance of a particular task. So to sum it up, explanations in neuroscience attempt to identify components and their functions. But I think that empirical results are putting pressure on that, or at least the generality of that approach. Some of the empirical results include the reuse of local regions in multiple situations, dynamic neuromodulation, interlevel interactions, context-dependent functional attributions, nonlinear interactions between evoked and intrinsic activity. All these complications suggest that this sort of simple mapping model is probably not going to be adequate to neuroscience going forward, right? So again, as I say here more generally, right, emerging approaches are more consistent with complex dynamic systems theory than they are with simple mapping or simple componential mechanistic explanation. And I suggest that in dynamic systems theory, the goal is less about function than it is about understanding the parameters and constraints that govern the behavior of the system. Right, so this, as it were, gives us a different way to pursue neuroscientific investigation, right? And I think this is important. Right, we should be thinking about what it is, what are the parameters, and again, this relates to some of the earlier stories. What are the causes that shape a system's trajectory through some kind of high level? So, I think this is almost my last slide here. I think that a better understanding of the notion of constraint and how constraint operates in the brain promises a better understanding of a kind of explanation in the sciences. So this is a descriptive project. But I think it also leads to the development of better tools for scientists for understanding complex formative project. This is what we should be doing instead of continuing to look for functional mapping. Again, so this is just that slide again. These ideas are spelled out in quite a bit more detail in these places in the book, in a case study of Starburst Amacrine cells, and then in a recently article understanding how we should think about the relationship between evoked and intrinsic activity in the brain. And I think that's it. So thank you.